all, I want to thank the organizers for giving me the opportunity to present our data. Certainly a great pleasure and pressure. While we heard about RNase P, we heard about the reaction it catalyzes. We also heard about that RNase P is considered to be an ubiquitous ribozyme. But we know at least one organism that doesn't need an RNase P activity, although it's a parasitic organism. And in the last 20 years, some positive RNase-free RNase P enzymes have been characterized. Among those, mitochondrial RNase P from humans. And to finally solve the question about the existence of an RNA-free RNase P, we focused our studies on human mitochondrial RNase P. Human mitochondrial RNase P turned out to be extremely unstable during protein purification. Therefore, we used an alternative strategy which was based on only partial purification. We generated 5 different partial purifications and shown in here. The partial purifications were in depth characterized by mass spectrometry, and we identified the subproteomes, and we identified between 166 and 362 proteins in those 5 different 2-step partial purifications. The next step, we compiled the overlap proteome, because in line with our rationale, we thought that the overlap proteome should contain RNase P components. Well, to our surprise, we only identified 24 protein components in 5 out of 5 preparations, which was astonishingly small. We identified more than 1,000 proteins in those 5 different 2-step partial purifications. We said RNase P components might be not enriched enough that we can detect it in our 5 preparations. Therefore, we took the 3+ overlap proteome, the proteins identified in 5 out of 5 preparations, in 4 out of 5 preparations, and in 3 out of 5 preparations, subjected to a bioinformatics screen for candidate proteins. We selected 3 candidate proteins. We overexpressed them in 293 hex cells and subsequently affinity purified them. And we saw that candidate protein number 1 was indeed associated with human mitochondrial RNase P activity, as shown in this activity assay, where the other 2 candidate proteins were not. And the further characterization of the candidate protein number 1 led to the identification of 2 more components of human mitochondrial RNase P, basically by co-purification. All 3 proteins were recombinantly expressed in E. coli and purified to homogeneity, and we called them MRPP1, 2, and 3 for mitochondrial RNase P proteins. And indeed, we were able to reconstitute human mitochondrial RNase P activity from those 3 recombinantly expressed proteins, as shown with 2 different substrates, mitochondrial pre-tRNA tyrosine and mitochondrial pre-tRNA isotype. Human mitochondrial RNase P, the recombinant enzyme, cleaves a variety of mitochondrial pre-tRNA substrates, and only those 2 are shown here. And by using proteins expressed in E. coli, we can definitely exclude the possibility that there is any human RNA involved in catalysis, especially not the H1 RNA. To exclude the very unlikely possibility that an RNA component of the expression host is involved in catalysis, we treated recombinant mitochondrial RNase P with micrococcal nuclease, which led to the complete degradation of the co-purifying nucleic acid without altering the mitochondrial RNase P activity. And to finally eliminate all doubt, we added M1 RNA to the recombinant mitochondrial RNase P, and we saw an inhibition of the reaction instead of the stimulation of the reaction. And the most prominent, the inhibition of the reaction could be relieved by completely digesting the M1 RNA, again with micrococcal nuclease. In summary, these experiments clearly show that there is no RNA involved in catalysis, and human mitochondrial RNase P is completely solidly composed of proteins, and there is no transacting RNA needed. Some further properties of human mitochondrial RNase P, like the native mitochondrial RNase P, and recombinant human mitochondrial RNase P is magnesium dependent. It cleaves at the precise 5' terminal of mitochondrial tRNAs. It produces 5' phosphates and 3' hydroxyls, which are all hallmarks of RNase P. Now, to make sure that those three proteins are also involved in 5' pre-tRNA processing in vivo, we knocked down the individual subunits by RNAi and measured mitochondrial pre-tRNAs with the quantitative PCR assay. As you see here, knockdown of MRPP1 led to approximately 30-fold accumulation of mitochondrial pre-tRNA valine. Knockdown of MRPP2 led to approximately 60-fold accumulation of mitochondrial pre-tRNA valine, and knockdown of MRPP3 led to approximately 6 to 7-fold accumulation of mitochondrial pre-tRNA valine. Now we measured between 8 and 10 mitochondrial pre-tRNAs, and only mitochondrial pre-tRNA valine is shown here because this was the one which accumulated the most. To also show that the pre-tRNA accumulation is due to the specific decrease in mitochondrial RNase P activity, we isolated mitochondria of RNA-treated cells at the indicated time points and performed an in vitro RNase P assay. And as you can clearly see, again, the assay was performed with two different substrates, tyrosine and isoleucine. And as you can clearly see that upon knockdown of MRPP1 and MRPP2, which leads to a strong accumulation of the mitochondrial precursors, we weren't able to detect any mitochondrial RNase P activity at all, whereas the control tRNA transfected cells or the untransfected cells are completely normal with respect to their RNase P activity. And the nice thing is, knockdown of MRPP2 led only to a 6-fold increase of the precursors, and you see a little bit of RNase P activity left here, but still it's very much decreased compared to untransfected and controlled tRNA. In summary, we can conclude that human mitochondrial tRNA 5' end processing does not require a trans-acting RNA component. The components of human mitochondrial RNase P are not homologous to the hereditary non-RNase P components, and human mitochondrial RNase P is a novel type of a patchwork enzyme composed of proteins recruited from at least two different metabolic pathways. MRPP1 is still involved in tRNA methylation, and MRPP2 is still involved in fatty acid oxidation, and MRPP3 is of unknown evolutionary origin. Last but not least, I would like to acknowledge some people. First of all, my PhD supervisor, Walter Rossmanith, is the technician in our lab, Peter Frank and Christopher Gellner from the Cancer Institute, and Kerry Bennett from Seminars.